the phage 174. Phage 174 is also a kind of bacteriophage to be present. Like M13 bacteriophage, which is going to consist of single stranded DNA viruses in the circular manner. Like that, a phage is also going to be consists of DNA viruses as single stranded. It is going to consist of 5386 nucleotides. Among them, 11 genes are going to code for the proteins, and 8 genes are going to code for the viral morphogenesis, which means for the assembly of genetic material inside the capsid need this 8 genes for synthesis of viral morphogenic proteins which involved in viral morphogenesis. Among this, a 44% is going to carries a nucleotides of GC content, which is going to provide a tertiary structure, and 95% of nucleotides are going to consist of coding genes. It is a GC content is similar to a introns, which is not going to code for any of the proteins. Next, we are going to see about the phage 174 structure. A phage 174 structure is going to consist of a spike protein, major spike protein and minor spike proteins are also going to be present. A spike proteins and capsid proteins are more in the phage 174, therefore it needs viral morphogenesis process as a major process to be present during the replication process, as it was a single stranded DNA genome. So here we can find the gene proteins. It is a single stranded DNA genome. It is going to be segmented as A star based on its letters. A gene which is going to be marked as A is going to have a replication factor DNA, a nick replication factor DNA, as we seen before the M13 phage, which is going to has a replication endonuclease synthesis by G2 gene. Likewise, a replication endonuclease is going to be synthesis by this phage by the A gene, and it also synthesis A star. This gene is going to inhibits the DNA replication to carry out the replication of the phage 174. Also infects the E. coli. Like M13, a host of this phage 174 is also a E. coli. It's going to inhibit the DNA replication of the E. coli to carry out the replication of the phage. As we said, for the viral morphogenesis, about 8 genes are involved in the viral morphogenesis. These genes are synthesized by B gene. These genes are B genes. It is going to synthesize the scaffold protein. Scaffold protein is going to act as a template for the capsid assembly. For the capsid assembly, a scaffold protein acts as a template to formation of capsid. Therefore, it is involved in capsid assembly. A C gene is going to involve in DNA packaging. After the capsid assembly, a DNA must be packed into the capsid. It is going to be carried out by the C gene. D gene is also involved in procaspid assembly. That means after the DNA is packed, outer capsid is going to be folded off with the scaffolding protein synthesis by the D gene. Then E is going to be a host cell lysis. As we see that both M13 and phage is going to infect the E. coli, but M13 is going to carry out the non-lytic cycle, where the phage 174 is going to carry out the lytic cycle, which causes the lysis of E. coli. Therefore, E gene is going to synthesize the host cell lysis, a protein to lyse the host, that is E. coli. F gene is going to produce the capsid proteins, and G gene is going to synthesize spike proteins, and H is going to synthesize minus spike proteins. These are all the capsids. And J is going to synthesize a protein that is going to bind to the single stranded phage DNA, and it causes it to DNA packaging. DNA packaging is carried out by C gene protein, and K gene is going to involve in optimizing the bust size. It going to produce certain protein that involve in optimizing the bust size, which causes the lysis at the particular size of the E. coli. Next, we are going to see about the replication of the looped rolling circle model of replication. Looped rolling circle model of replication. From the name itself, we can find that there is a rolling circle model of replication like M13 phage, but there is a looping to be taken place during the rolling circle model of replication. So, this is going to be a DNA strand, single stranded DNA strand. When enters into the E. coli, it converted into a double stranded DNA strand. After it becomes a double stranded DNA strand, a gene A protein is going to be attached. After it converted into a double stranded DNA strand, a positive strand is going to be a strand of phage, where the negative strand is going to be a replica of it, which is going to be formed for the double stranded structure, where a nick is going to be made in this positive strand by endonuclease enzyme similar to the M13 phage replication. When there is a nick was made, a gene A protein is added. This gene A protein get attached to the 5-phosphate like a M13 phage, where it gets attached to the plasma membrane of E. coli at the 5', where in the 5' here the gene A protein get attached. After the gene A protein is get attached, a replication is going to be carried out, where a rolling of the negative strand is going to be taken place, and there is a synthesis of a positive strand to be taken place. A newly formed strand is going to be a positive strand to be synthesized continuously. As leading strand is going to be synthesized. Only difference is in the M13 phage, a leading strand is going to be synthesized at the 3'N, and the lagging strand is going to be synthesized at the 5'N. But here in the phage 174, a leading strand is only synthesized. No lagging strand is going to be synthesized. To prevent it from the lagging strand synthesis, a SSBB protein is going to be attached to these 5'N, that is positive strand is going to be attached with the SSBB proteins and the gene A protein, where a negative strand, a duplex to be formed, is going to involve in the newly formed DNA. A newly formed leading strand synthesis is going to be taken place. At first, the RNA primer is going to be added and continuous synthesis to be taken place till the replication meets the origin. After the origin is meted off, a gene A protein is get attached to the newly formed DNA 5' ring. 5' end of the newly formed DNA is going to be attached to this gene A protein. Here we can find that a positive strand, a old positive strand, and a newly formed positive strand. 5' end. Both 5' ends are get attached to the gene A protein. After that, it's going to be released off. Therefore, in the positive strand, a gene A protein, a newly formed positive strand where the gene A protein get attached and the already entered the positive strand is going to be released from the gene A protein and it was formed. Therefore, this cycle is going to be repeated off. Therefore, here the looping is going to be occurs by the SSBP protein and gene A protein, and a rolling back is going to be occurs. They looped rolling circle model of replication. So, a looped rolling circle model of replication is going to generate a progeny of single-stranded circular DNA. It's going to generate the replication in the single-stranded circular DNA, especially in the phase 174. This replication is going to be taken place. Here, a gene A protein is going to be an important protein to be taken place. It's going to attach to the 5' phosphate terminus by covalent linkage. Using a replication protein, an SSBP protein, it's going to be stopped from lagging strand synthesis, and a displacement of broken parental strand is going to be taken place and continuous rolling back to be occurs. This rolling back causes us to loop of the 5' strand, that is positive strand of 5' end. This strand becomes coated with SSBP proteins and not replicated, where the leading strand is continuously replicated. A gene A protein is now bound to the 5' end of a positive strand, and then a original nicking event is going to be taken place. It leads to dissociation of the positive strand. Thus, a positive strand is going to be dissociated, which is attached to SSBP protein. A old positive strand is dissociated and the new positive strand is going to be attached with the gene A protein, that is replication protein, is going to be attached off. It causes the circulation of the strand as a single stranded circular DNA is going to be synthesis. Then the cycle is going to be taken place till it reaches the optimum amount of DNA for the progeny, for the daughter viruses.